All right. Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the three national centers established by the NIH Common Fund Transformative High-Resolution Cryo-Electron Microscopy Program. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. With me today from NCCAT is Ed Eng. And joining from the Pacific Northwest Cryo-EM Center, PNCC, is Kirsten Wheeler. And from the Stanford SLAC Cryo-EM Center, S2C2, is Michael Schmid. S2C2 has invited today's speaker, Professor Eric Greene, who's joining us from San Francisco State University. And Mike will introduce him properly after a quick update from each of the three centers to keep you up to date on the resources we make available at no cost to the research community. This webinar is a regular event we host the last Thursday of each month, highlighting the methods and tips and tricks that groups are using to collect and interpret the data they can obtain with the help of National Center resources. You can save the date already for our next talk, which will be on February 29th. We get a whole extra day on the calendar this year. So we're excited. We get to spend 1 hour of that day with all of you thinking about how to use cryo-EM to study exciting biological questions. The speaker and talk title are coming soon, and you can look out for that information as well as registration links on the events page at cryoemcenters.org. One more announcement from me before we kick off. If you're going to be in Philadelphia in a couple of weeks at the Biophysical Society annual meeting, come say hi to us in person. We have a booth number 101 in the exhibit hall. That's to make us easier for you to find. And staff from all three centers and also the cryo-electron tomography centers will be on hand to answer questions, chat about all things related to using National Center resources, and any other cryo questions you may have that you want to come talk to us about. So find us at the booth, and if not at the booth, as usual, be on the lookout for Ed Eng with the NCCAT chameleon around the conference. So back to today's business. One final detail, if you have questions during the talk, you can send them using the Q&A box. Please send them using the Q&A and not the chat, and Mike will be moderating questions at the end of the talk. So now we'll turn over to a quick update from the centers. Ed at NCCAT, you can take it away first.
Hi, I'm Ed from NCCAT. We're here in New York City. We offer three main categories of access: high-end instrumentation access, a robust screening program, and a cross-training program. Something to mention that's coming up, this is conference season. A lot of abstracts are due. In microscopy and microanalysis in July in Cleveland, we are hosting a couple of symposia. So if you're ready to go to a conference this summer, we suggest that you submit to B04, Electron Microscopy and Education. If you have a more general topic, you can submit to B01, 3D structures from macromolecular assemblies to whole cells. So I urge you to apply because in February, the deadline is due for M&M abstracts. If you want to find out more, please go to nccat.nysbc.org.
Thank you, Ed. Kirsten, I'll let you go next.
Hi, my name is Kirsten Wheeler, and I'm the Administrative Coordinator for Pacific Northwest Cryo-EM Center. PNCC has two types of proposals. One is our screening access proposal, which allows one user to come on site for up to 5 days of sample preparation and screening with staff. This proposal is aimed at low resource institutions. The second proposal type is general access, which awards up to 480 hours of microscope time a year for 2 years. This time can be used for screening or data collection, as well as the fact that general access proposals are eligible for our training workshops, which cover topics from sample preparation all the way through to data processing. PNCC has 5 microscopes. We have an Arctica with a K3 camera for screening and 4 Krios microscopes for data collection equipped with K3 and Falcon 4i cameras. 3 of our Krioses are equipped with Glutamine energy filters. For sample preparation, we have 2 Vitrobots, a Leica GB2, and a Vitrojet, which is still undergoing validation before being offered to users. We are expecting the arrival of a new Glacios 2, which is actually happening next week. We are very excited about that. Lots to be done. If interested, please go to pncc.labworks.org and apply today. Deadlines for proposals are the 1st of each month.
Thank you, Kirsten. And now I'll turn over to Mike.
Hi, I'm Mike Schmid. I'm one of the directors of the Stanford SLAC Cryo-EM Center, S2C2, and we have 4 Krios microscopes available and a 200 kilovolt microscope available. We have, as the other 2 centers, we have proposals for high resolution data collection, for screening, and for training. And we also offer workshops. There will be one in February where users can test drive our new Chameleon and Vitrojets, and of course the Vitrobots that we already have. And we expect to have users on site and lectures for people who want to do it to learn about them remotely, including screening right after the freezing. So that's going to be very interesting. Then later in the year we'll have image processing workshops, and every week we have a roundtable for people who want to ask questions about data processing as they work with the data they collected here. See us at S2C2, and we hope to see you.
All right, so I'll stop sharing. And Mike, now you can introduce today's speaker.
This month's speaker, we're very happy to have Eric Greene, who got his undergraduate degree at University of Colorado at Boulder, and then did graduate studies in the Bay Area at UC Berkeley, and postdoc work at UC San Francisco. He is now an assistant professor at San Francisco State University, perfectly located in between all these great places. It's a primarily undergraduate institution, but it's really well situated for people to come and use our facilities and the other facilities in the Bay Area. I drive past San Francisco State on my way up to cross the Golden Gate Bridge, and it's a very nice place, except for the traffic on 19th Avenue. And it's great to have Eric here. He's had 2 projects here, one training project, and then a high resolution data collection project. We're very happy that he's made use of our facilities, and I heard him talk at the SSRL user meeting last fall, and he's done great work, and we're very happy to have him represent S2C2 and his own work here. Go ahead, Eric.
Mike, thank you so much for the invitation and the really kind introduction. I'm really excited to be here to share this work with you all. This is part of it is my postdoc work, and part of it is now carrying on into my own independent lab now at San Francisco State. I entitled this "Decoding the Complexity of Human Glutamine Synthetase" because I think though this has been a model enzyme system for many decades, I think with new technologies in cryo-EM, and specifically looking at things from an ensemble perspective, we're able to really gain new insights into this fascinating enzyme. So I think before I get into glutamine synthetase, I just want to talk first about some of the overall motivations for me as an enzymologist. I come from a protein biochemistry background. Yeah, protein sequences themselves encode all of the information needed for their function, right? So the primary amino acid sequence dictates the 3-dimensional fold that an enzyme adopts, where the active site residues are positioned to make an active site. But what's been more and more appreciated in recent decades is that in addition to that, the amino acid sequence also encodes the energy landscape. So all of the available conformations that are available to a protein to allow it to undergo catalysis. And so these are just a couple recent examples of some very small conformational changes that drive catalysis, as well as large ones for adenylate kinase. And I think one of the areas that is needed in cryo-EM studies is to really look at this dynamics under catalysis and to understand how these play a role in actually facilitating what enzymes do. And one thing that's really great, so cryo-EM has a bit of a size limitation. You can only go so small, but most enzymes actually exist as oligomers. And these are the 10 enzymes of glycolysis from the PDB's molecule of the month. And this is just a subset of the different oligomeric states that these can form. I think it's very interesting that there are different oligomeric states depending on different organisms. So I think that there's a lot of room to continue studying these enzymes and looking in more detail at their structure, and especially their structure while undergoing turnover. But coming back to glutamine synthetase, it's a bit of a ubiquitous enzyme. It's found in all domains of life, and it plays a major role in nitrogen fixation. So nitrogenase is the primary enzyme in the biosphere that fixes atmospheric nitrogen to ammonia. But once you have ammonia, really being able to utilize that to actually make different biomolecules is key, and glutamine synthetase plays a big role in that. The reaction carried out by glutamine synthetase, is, which I'm going to abbreviate from now on as GS, it phosphorylates glutamate to make a gamma-glutarol phosphate intermediate, and then utilizes ammonia to form glutamine through a SN2-based phase 2 reaction. And it's being ubiquitous across these domains of life. It's considered an ancient enzyme, so it was around the time of the last universal common ancestor. And it also has a bit of a unique architecture. So what I'm displaying here is human GS, which is a dimer of pentamers in its canonical oligomeric state. Its active site is actually formed at the interface of two monomeric units. You can consider it a bifunnel shape, where ATP will bind one side, glutamate and ammonia will bind the other, in order to form the full complex to undergo catalysis. And in eukaryotes, we're less concerned about nitrogen fixation in the biosphere. We're more concerned about amino acid homeostasis in the body. And so one thing that I found very fun to now be teaching biochemistry is to actually learn a little bit more deeply about metabolism in cells. And amino acids are actually the major metabolite in eukaryotic cells. This is from a recent publication. Amino acids are over 50% of the total metabolite contribution for human cells as well as yeast. These are used, of course, for the building blocks of proteins, but also can feed into a TCA cycle, for instance, to help in energy production, as well as be utilized for other molecules like nucleotides. And so the body really has to balance this biosynthesis and catabolism, meaning that any enzyme that's involved in amino acid homeostasis really needs to be under tight regulation. And glutamine is the most abundant amino acid in our bodies. Being able to carry that extra nitrogen group on this amino acid, it can then donate that to various processes like nucleotide biosynthesis. I mentioned how it can feed a Krebs cycle, but it's also involved in glutathione biosynthesis and neurotransmitter homeostasis. So being able to regulate the levels of glutamine in our body is really paramount to leading healthy lives. And glutamine metabolism is, of course, implicated heavily in cancer. And the only enzyme in our body that can actually synthesize glutamine is glutamine synthetase. So let's jump in. We're just going to present a little bit of an outline of my talk. So it's going to be in 3 different chunks. The first is going to be a story about product stabilized filamentation and how this negatively regulates human glutamine synthetase activity. The second is going to be about an ensemble function analysis, looking at an allosteric network that's connected to some of these oligomeric interfaces. And then the last section is going to be a really fun time that I had in my first semester, creating a course-based undergraduate research experience as part of the upper division biochemistry lab that I taught, looking at disease variants of human glutamine synthetase. But let's dig into this regulation. So bacterial glutamine synthetase is a great model system that was really worked out many decades ago for just kind of an archetype of negative regulation. There's multiple factors that perform feedback regulation of metabolites, as well as an intricate post-translational modification system that can all very exquisitely and very quickly turn up or turn down the activity of bacterial GS. By contrast, the literature for human GS is a little sparse. We know that as glutamine forms in the cell, this will actually trigger a degradative process, so that will install acetylation marks as well as ubiquitin marks to degrade GS in the cell. Where feedback inhibition and post-translational modifications are very fast over here, this whole degradation process is very slow. This is like a multiple hours to 12 plus hours phenomenon. And so one of our questions that we wanted to answer is what regulates human GS activity quickly, like on a fast time scale that is needed for the cell to actually adapt to changing nutrient conditions. And so one of our hypotheses, and this was also based on some previous literature that has suggested that glutamine synthetase was able to form filaments, would be that it could be regulated by filament formation. So over half of metabolic enzymes, it's predicted, can form some sort of filamentous structure inside the cells. And a lot of work, especially spearheaded by labs like Justin Coleman up in University of Washington, have really uncovered a huge amount of different regulatory mechanisms that can be applied to metabolic enzymes to regulate their activity based on filament formation. Some of them can cause conformational stability, can cooperative switches, can turn on or off the enzyme based on whether it's in a different filament and how it reacts with different modulators. So this was predicted for glutamine synthetase, but no one had been able to show it before. And the other important aspect of metabolic enzyme filamentation is that it's fast. So you don't need to synthesize or degrade any new proteins. It's all the same proteins that are in the cell that can come together, fall apart, change their conformation all very quickly to tune activity. So when we first started this project, we had crystal structures available of glutamine synthetase, and they all had N-terminal his tags still on there. And previous work from a grad student that was in my postdoc lab has shown that his tag really isn't so great for being able to actually have the full function structure of glutamine synthetase. So when we have the his tag, this is just a 2D class average from a negative stain EM data set. It's very unstable, and these two pentameric rings here you can see kind of flay open like a butterfly. So it was really necessary to get a scarless preparation set up for this enzyme. When we have no more his tag here, we can get nice planar rings that have high 2D class average resolution when we then move it into cryo-EM. And this is just a cautionary tale about his tags. So if we have N-terminal his tags and our N-terminus is projected into the center of the molecule like this, if I actually ran the numbers, and it was kind of interesting, if you have 10 for a decamer his tags all projecting into the center of this complex, that has the same molecular weight as GFP all stuffed into the center of an enzyme, that it could form a decamer at all, I think is quite surprising. And importantly, the his tagged enzyme was about 10-fold less active than its tagless counterpart. So this is a cautionary tale about watching where your tags are, and then also just to ensure that you have a really good protein system before starting structural studies. And I think I went into this admittedly a little naively. I was just very interested in trying to capture what enzymes look like when they're undergoing catalysis. So cryo-EM has this great benefit where you can freeze samples in vitreous ice in near native-like conditions, and they're stuck in time. So you can add substrates to your enzyme of interest, react for a certain amount of time, and then quench freeze on grid to stop whatever processes might be happening. And so I was very excited on like my third EM experiment. This was at UCSF. When I did this, I found that we actually saw filamentation right on the grid. Each one of these little black lines is an individual pentamer of glutamine synthetase. Sometimes you can see these five-fold star structures, but you can see that there are filaments that formed directly on the grid. And so we were very excited to see this. And as I mentioned before, numerous metabolic enzymes form these higher order structures. This is a great study that screened many enzymes in yeast with GFP tagged libraries and found that about 60 of the 116 total that they screened could form some sort of filamentous-like structure in yeast. And indeed, in S-VCA, there's GS forming a puncta that then we're following up with the in vivo studies. And this was a repeat experiment that was conducted at SLAC during my training proposal. And I was very happy to see that this could repeat itself. And not only that, but occasionally you could get these really long filaments, which was really fun to see. Another, of course, real powerful application of cryo-EM is that you can really tease apart different compositions. So luckily, most of the filamentous particles in my micrograph were very discrete in their size. So a lot of them happen to be a dimer of decamers now. So we could actually separate out these particles through 2D class averaging. And then take these particle sets into a homogeneous refinement to get very high resolution structures of around 2 angstroms. I was very, very happy. I was at Biophysical Society 2022, and I'll also be at Biophysical Society 2024 in Philadelphia. So if anyone wants to come talk to me about this research or anything else, I'm happy to talk there. It's a great meeting. I've been going to it for a few years now, and I was very happy when I was at Biophysical Society 22 when I actually got to meet Justin Coleman, and he came up to me. He said, "My student has a fibril structure of your enzyme. We should talk more." And I was like, "Oh." I was a little scared at first. The Coleman lab has solved just a ton of different cryo-EM structures of enzymes and is really at the forefront of this field. This is just a small subset of some of their recent studies. But luckily, we wanted to collaborate with each other. And so now I've been working with this very talented grad student, Richard, who provided an APO form, glutamine synthetase, filament structure, also at about 2 angstroms. And so we've been working with that to look at what are some of the conformational differences that are occurring upon filamentation and what is this actually doing. So the filament formation itself for GS is this head-on-head arrangement. So we have a canonical decamer and a second canonical decamer that go head-on-head. And there's 5 different small interfaces that are formed. And when we looked at each small interface, we were just a little surprised that we didn't really see too much density in our EM maps that really bridged this. It was very stable. These structures could appear in multiple different data sets under different conditions, so we believed it to be there. But we didn't really see very compelling density between it. So I wanted to biochemically verify that this was in fact the interface, was in fact a bona fide interface. And so luckily, by having a fully modeled filament structure, I did some distance measurements and found that the only lysines that were across monomers and in close spatial proximity where the lysines contributed at this interface, lysine 52, which faces the other lysine 52. And similarly, the only 2 cysteines that are across monomers in the system is cysteine 53. This is just one example of a cross-linking experiment. I used a bifunctional NHS ester to cross-link K52 to K52. This had a very, very short spacer arm of 7.7 angstroms, so it's very restricted. And what I could see was that in our wild-type enzyme, I could get pretty robust cross-linking across this interface. Mutation of that lysine to an alanine mutation, basically completely ablated that cross-linking, and so did the other cysteine 53 to alanine mutation, also obliterated that. I could also do the same experiment with maleimide bismaleimide 8-angstrom crosslinker and get the same results. And so we were very happy that we could indeed biochemically verify that this was a bona fide interface. The next step came when we wanted to compare the turnover filament structure that I'd shown you earlier with the apofilament structure. And one thing that we saw when we started comparing these was that there was actually this nice big blob of density that bridged these two monomers at this filament interface. And so we started thinking like, well, okay, we have two different cryo-EM experiments, one in apo conditions where we didn't have any substrates, and then one in turnover conditions. So it's a very defined experiment. We know exactly all the chemical compounds that are in there. So what could be fitting in here to make this work? Because we knew that none of the amino acids could really form this density on their own. So we could rule out first all of the things that were too small or too big and things that were the same between the different experiments, which left us with just two molecules that it could be. It could either be the substrate glutamate or the product glutamine. And so this was a really fun experiment to do. I think one of my favorite results that I've seen is I wanted to do a time-resolved cryo-EM experiment to look for whether or not it was glutamate at early time points in the reaction that was seeding this filament formation, or if it was glutamine. So how I did this was I took 5 different data sets at Slack, each of these corresponding to a different time point in the reaction from the time that it was initiated. So I would initiate my reaction in an Eppendorf tube right by the Vitrobot, and then I would apply it to the grid and record the time that it was sponged as the quench time. I initially started with taking each one of these data sets individually, classifying it out in 2D class averages as decamer and filament, and then taking this fraction of the total particles and plotting it against time. And what was just so validating from this experiment was that we could see that the decamer particles went down linearly with time, and the filament particles went up linearly with time, thus showing to us that glutamine formation by the enzyme was actually helping to stabilize this interface. And so now, in a subsequent experiment, I did just ATP and glutamine, and was also able to see filament formation in micrographs. This is just some screening data on the Arctica to confirm that. And so we can now rule out glutamate, rule in glutamine, and also model glutamine into this density. And so, so our model now is that filament formation can form by 2 different mechanisms. One is overall enzyme concentration. So for my cross-linking gel that I showed earlier, you can see there's more filament formation at the higher concentrations. But in addition to that, filament formation can also be stabilized by the product of the reaction, glutamine. So next we wanted to see what is the functional consequences of filamentation for catalysis. And so knowing that we could stabilize filament forms, and we knew also the concentration regime that the enzyme needed to be in to form these filaments, I did many steady state experiments, and it took a while to understand that it was actually just the Km for ammonia specifically that was altered upon filamentation. So what we could do is we could do steady state kinetics with wild type or wild type plus a saturating concentration of glutamine, compare the Michaelis-Menten constants for this. And what we could find was that there was little or no change to Kcat, but that there was about a 3-fold change to Km, and specifically for ammonia, there was no Km changes for ATP or glutamate. And this is actually quite important that it's specific to ammonia. So, and I just want to contextualize this result for a second, which is that in our micrographs, at most, like 50% of the particles are in a filamentous form. So I should say that we are not fully forming filaments here, and we're not fully not forming filaments here. So this difference is actually an underestimate of what the likely difference will be for specifically filament and specifically non-filament particles. Additionally, the physiological concentration of ammonia in the cell is around 150 micromolar. So this is actually pretty substantial. This would mean that in a no glutamine context, about 85% of GS is undergoing forward catalysis here. And if glutamine is present in stabilizing filaments, that only about 50% of particles are undergoing catalysis here. So this is really hitting at the heart of what physiological regulation could look like. And so next we wanted to ask, like, okay, we have now a functional difference in ammonia cam. So like, what is actually happening at these active sites that could be eliciting this effect? So I mentioned earlier in the talk that the active site forms a bifunnel-like shape. And we actually had crystal structures that we could look back to of this enzyme from previously in the literature. Which really suggested that we should be looking at this E305 loop. So there's two crystal structures of human GS, one of them in just a ADP condition where an active site loop is disordered and not modeled, and then one with a transition state inhibitor included, where this E305 loop is very well ordered and closes over that glutamate and ammonia binding pocket to really help facilitate catalysis. And this pocket here is, also helps regulate ammonia entry too. So when we started looking at the E305 loop, we started also seeing differences in the density for this loop in our different reconstructions. So I'll take you through this one by one. First, in our apofilament map, we notice very strong density for this loop all the way across. And then if we look at our turnover decamer, I'll go across over here. We see that the density is fairly eroded, that it seems to be sometimes in this closed conformation, I'll call it, and sometimes not. This looks similar to an ATP containing filament structure that was also provided by Richard. And then if we contrast all these two, where we have partial loop density to the turnover filament, we see that we lost a lot of the density for this loop, and that it's nearly gone here. And so we next wanted to ask the question, we can see density for the loop in this closed ground state, but what does the no density, what could that look like? And so we turned to some friends of the Institut Pasteur, Max Bonomi and his talented postdoc Sam Hoff, and we did molecular dynamics ensemble refinement into these different maps. So how this works is it's both the molecular dynamics simulation, so it takes in physiochemical force fields, but then it also takes in information from the cryo-EM map itself to guide the refinement. And so the ensemble refinement here really revealed that, well, one, the active site loop is very variable, so even in conformational state. So this is an overlay of all of the different ensemble models that were here, which is close to 6,000, and showing the conformational space of the active site loop in the decamer turnover condition and the filament turnover condition. So even in the partial loop density that we could see, the active site loop can adopt many conformations, but in the low to no density that we saw in filament formation, this loop adopts many conformations all over the place. It has a very wide distribution of conformations that it can attain. So we started digging into this a little deeper. We wanted to understand what were some of the key interactions that would really stabilize that closed conformation of the loop. One that stood out to us was that there was a small hydrophobic pocket that was formed from leucine 300, isoleucine 285, and isoleucine 309. What I'm showing here is like a ground state model for the two systems. The decamer is in magenta and the filament is in blue. In this decamer state, we can see that this forms a nice hydrophobic pocket here, but in the filament state, 309 and 300 break away from this hydrophobic pocket and can be seen elsewhere. If we look at the full ensemble, Here, turnover decamer is in orange and turnover filament is in blue. We can see that the turnover decamer, there's a lot closer distance between leucine 300 and isoleucine 285 in the ensemble. However, the turnover filament, this entire distribution is rightward shifted, and this interaction is pretty rarely formed. So we did a lot of mutagenesis studies here with these different loop interactions. We could find that an L300A mutation could have a KM for glutamate that was about 100x wild type. So if we disrupt this pocket through mutagenesis, we can have a really big impact on its ability to form a Michaelis complex. Another interaction that was pretty key that we could see was a salt bridge that formed between R298 and D239. This is the same color scheme that I showed before with the decamer ground state in magenta and the filament ground state in blue. In blue, we can see a nice salt bridge that's forming there, or in magenta, excuse me, nice salt bridge that's forming there. However, in the filament state, that R298A is actually very far away. Similar result that we can see when we look at the full ensemble. The salt bridge can form sometimes in the filament, but a lot of times it's broken, and either mutation of R298 to A or D239A, D239 to A also caused a very large 50-fold increase in KM for glutamate here. So these positions are highly sensitive to mutation and also pretty well conserved. To just confirm that we were really interrupting the ability of the loop to form this active closed state, we got a structure of the R298A decamer under turnover conditions. This was also a very high resolution structure. And what I hope you can appreciate even from just this single representation of the EM density is that you can't even see these active site loops that would be about here whatsoever. And if we zoom into it and even give the density a really high threshold, such that you can really see a lot of the side chains coming off here, we really see nothing for the loop that's modeled here. So disruption of the salt bridge really does just allow very high conformational heterogeneity for the loop, and that is part of what's driving the kinetic defects that we're seeing. And so then we wanted to understand, like, what is driving, or what could be contributing to this loop being in its closed ground state versus allowing it to adopt all of these other conformational states that it could arise. So it took a little while to really try to zoom in on what the conformational change is in the system. And so this is a very complicated figure that I'll work through part by part. So first, dark blue and dark green are going to be our filament ground state, chain A and chain B. In cyan is going to be the decamer chain B, and in lime is going to be the decamer chain A. Aligning the model on chain A here, so we can see that there's high alignment on this side. We can see that between the decamer and the filament, there's a conformational change right at the interface between these two monomers. So right behind these polypeptides is the active site. Down here is the E305 loop in its very conformations. And what we can see is that in the filament, the minor subunit, so there's a major subunit constitutes most of the active site, a minor subunit constitutes a small part of the active site. There's this conformational change away from the active site, so from lime to green here. And one thing that could be important for this conformational change is E305, which is the residue right at the very tip of the loop, participates in catalysis partially by deprotonating ammonium ions to ammonia and directly contacts residues on the adjacent monomer here. So we think that even subtle changes in the distance here can disrupt those interactions, also disrupt the ammonia binding site, and could be contributing towards allowing higher conformational freedom to this active site loop, and is what is our current structural hypothesis for how the ammonia KM is disrupted in the filament formation itself. So our overall model for the conformational landscape of GS under catalysis is we can have our active site loop. Upon ATP binding, there's a conformational change that induces disorder in the loop. And sometimes you can have very high disorder, such as conformations like this open twisted that we saw in the ensemble. If this conformational landscape is favored, then KMs for glutamate and ammonia are quite high. If you make any disrupting mutations to the loop itself, you'll promote this open, twisted, high KM state. However, if you're in wild type, you can find yourself back into an open state of the loop where it's kind of not twisted on itself, but it's unfurled like a flap, which is near its ground state in the closed conformation. Here, you can attain a low KM, so a tight Michaelis complex for glutamate, but you can still also have a high KM, low Michaelis complex for ammonia. And filamentation directly impacts the ability of the enzyme to form the active closed conformation. But glutamate binding can help stabilize this closed conformational state here. There are multiple residues on the loop that contact directly the substrate in the pocket here. And once you are able to form a closed active conformation of the loop, that'll help drive down the KM for ammonia finally to help facilitate efficient catalysis. And so how we want to contextualize this is as a conformational landscape is through an energy diagram. So we can see that, and how I presented this is a deep, low energy well for this enzyme. It's still in its 3D shape. The active site residues are largely very rigid and in place. So we're really looking at relatively small differences in total energy between these different conformational states that we can zoom into. And in an active decamer, we can see that it is slightly favoring an active closed loop, but can still also have an open ensemble during turnover. If we add a loop mutant, the open ensemble is definitely favored, and it has a much harder time forming the active closed loop conformation. And then finally, if we have filament formation driven either by concentration or glutamine addition to see that filament interface, we can see, or we present that it's going to be similar kind of low energy states for both an open ensemble and active closed state. So we can still do catalysis, but it's going to be a little hampered here. All right, so I hope I've been able to convince you that a product of the reaction can stabilize filamentation to negatively regulate human GS. And so now I want to move on to the next part of the talk, which is going to be an ensemble function analysis to look at specifically allosteric networks that are tied to these different oligomeric interfaces. And this is really driven by one question, which is, are the conformations present in the GS filament also present in an active decamer ensemble? And some of the inspiration for this came from just seeing some of the case studies actually for 3-key variability analysis from CryoSpark. So I should say in my PhD, I worked on the 26S proteasome. We were largely concerned with the 19S regulatory particle not pictured here. And we just kind of thought that the 20S proteasome was just like this brick. It was a protease that didn't really have much conformational changes. It certainly doesn't have the conformational changes of the 19S, but it's an EM standard too. So when we saw that you could see some variable conformations of the 20S proteasome, I just thought to myself, like, oh, that's very interesting. I wonder what kind of heterogeneity we could parse out from these, what otherwise looked like pretty rigid decameric composition GSs that we had. And so one thing that I was, so I started this project because we had a huge amount of data, and I wanted to start to mine that for what the conformational space would be. And I've been very fortunate to be working with Hiroki Yamamura. So he works for JSR in Japan. He's on kind of a sabbatical there as a scientist to learn cryo-EM with me. And so he's been with me for about a year and a half now, and has been the primary driver of this project. And so to start this, we just started with the very first data set that I presented at UCSF. It was a large data set, so we ended up with 1 million particles in a consensus map. With C1 symmetry applied, we had about a 2 angstrom resolution that we could attain with that. And so we fed this into the built-in 3D variability analysis in CryoSpark to just, honestly, we started a little naively just to see what we could see. And one thing that immediately stood out is that if we look at the 3 principal components from 3D VA, is that we can actually see loop density changes in all 3 of the principal components. And so this was, to me, like not a smoking gun, but it just really suggested to me that we should be following up on this further. And so we wanted to look at it through the ensemble function lens. And so we wanted to generate an ensemble. We took the 3D variability that we looked at, utilized the cluster analysis. So we broke this into 20 clusters based on where they are in the 3 principal components of the analysis. So each one of these clusters had about 26,000 to 113,000 particles. We did 20 individual homogeneous refinements after this. We applied C5 symmetry to this. As with many systems, we have preferred orientation issues, so the symmetry helped us alleviate that. Each one of these maps has a high resolution. And then we did a variability refinement in Phoenix to refine a seed model independently into all 20 maps. And then we used iSoldy to clean up these models. And what I've presented down here is a global RMSD. So this is a root mean square deviation for each one of the 20 models compared to the seed model from the consensus map. And the scale here is between 0 and 3 angstroms. So we see that there's very large sections that are very similar, and then some sections that are quite different. So now with our first, I call this the pilot ensemble. With our pilot ensemble, then we started to say, well, what kind of conformational states can we see? And how are these related to the loop? The movie that's playing on the right is just a random ordering of the 20 maps. One thing that I hope you can appreciate from this is that you can see a bit of a twisting motion between the 2 pentamers relative to each other, as well as you can see that there's different loop densities that are also corresponding. So we started to divide these up. We could say we did an ensemble analysis in Chimera to say like what rotational state are the different maps in, and then what is their loop density. And so we could find that there was one rotational state that had very low loop density, and then one rotational state that had that could support a higher loop density. So it seemed like there was from this analysis, it seemed like there was some sort of coupling between this conformational change at the pentamer interface to the loop itself. And so we started to investigate this pentamer interface a little bit more deeply. We could see that there was, in addition to a main interface that's formed between 2 monomers across the pentamer interface, we could also find that there was an alternate interface across. So we could kind of form a triangle between 3 monomers here to stabilize the pentamer interface. And when we started looking into our ensemble, specifically at residues from the pentamer to the loop, we could see that there was a few residues here near this alternate interface that seemed to have some variable conformations. And when we we looked at this manually just to look at which residues were had the most conformational difference, and we also looked at this. Some of my current students have looked at this through a correlation plus, which is more of a computational tool to define allosteric networks in an ensemble, and we were very gratified to see that our residue picks were very similar. And just to orient everyone in this picture, right, the active site entry is over here. This is the active site loop from the ensemble models. And this is our predicted allosteric pathway from the pentamer-pentamer interface up to the loop that was predicted by correlation plus. And so we started looking, so we wanted to see like, well, are these things actually coupled? I'm more of a biochemist, so I have to go to experiment. And so first we looked at the conservation. So what I've plotted here is a multiple sequence alignment. This is only of metazoan type 2 GSs, so it's a very small space that we're looking at, a small sequence space that we're looking at. These are the residues that constitute that main interface that I showed before. One of the residues that stood out to us as having variable conformations was this N152. Interestingly, it was not as conserved as the other residues in the interface, and there's a small population where this residue is glycine instead. And the other residue that we were interested in was H145, which also wasn't as conserved as other residues here, and had a significant proportion of sequences where it could be arginine. So we made the H145R and N152G mutations with the idea that we wanted to be as conservative as possible evolutionarily to see what sort of functional impacts interfering with this interface could have. So what we found for H145R, KCAT was also unaffected, similar to our previous studies. But we had a 2.5 and 3.5 x increase in KM for glutamate and ammonia. So that was pretty interesting that it seemed to be kind of on pathway towards some of the other KM changes that we'd seen before. For N152G, again, KCAT was unaffected. We had another KM of about 3-fold increase for glutamate. But then we saw something that was really peculiar. We actually couldn't get a KM for ammonia. We couldn't determine this. So what was going on there? And what we ended up finding was that for this variant, it was displaying a non-specific ATP hydrolysis, which was something that we hadn't encountered before. But basically, there was no additional ATP hydrolysis stimulation really from including ammonia in our reactions. So we decided to see, could it just hydrolyze ATP with only glutamate and ATP and not completing this full 2-phase reaction cycle? So we omitted ammonia here and only looked at the ADP that was produced with these two substrates. What we can see here, this is a coupled ATPase assay. So this is a loss of NADH, which absorbs at 340 nanometers. And what we can see is that under these conditions, where most variants show very little activity, little to no, because they require really forming glutamine to eject all of the substrates out and repeat the reaction cycle, N152G was decoupled and had robust ATPase activity without any ammonia present. These are some of the steady state rates for these various variants, and N152G is about 20-fold more active in this regard than other variants, and these other variants were about the same as wild type, which was measured in a separate experiment. So we were like, okay, this is very interesting. How do we get from this stable intermediate, which has been shown to be very stable by many studies, and through our own analyses, and how do we just end up losing this intermediate entirely? And we also wanted to see if we did include ammonia, how does this off pathway losing the intermediate compare to the on pathway forming glutamine? So one thing I did was I developed a steady state glutamine assay. So in one phase of the reaction that we can measure with by ADP that I showed previously, I wanted to then measure glutamine to look at the full reaction and get all of the steps here. And so this glutamine assay, I reconstituted a de novo NADH biosynthetic pathway with an ATP regeneration system. So it's 2 downstream enzymes that I added here to facilitate a biosynthesis of NADH. And so now we can, and these enzymes are in high enough concentration that they're not rate limiting. And part of the enzyme cycle could regenerate glutamate too. So we have glutamate regenerated and ATP regenerated. And so now we could measure steady state rates of glutamine formation over time. With this assay, what we found was that for N152G, that there's about the same rate for its non-specific ATP hydrolysis as its glutamine formation rate. By contrast, for wild type, this is about a 30-fold difference in these 2 rates. So this suggests that about 50% of the ATP hydrolysis events do lead to glutamine formation. And so this is a very decoupled, very inefficient enzyme from what should have been a very evolutionarily conservative mutation. So of course, having cryo-EM in our back pocket, we wanted to do that experiment. And N152 under turnover conditions, we did this on a glacios at UCSF. So it has a little bit lower of a resolution. One thing that we noticed was that there was an asymmetry between the 2 pentamers in our reconstruction. And so we're digging into this now. Our working model right now is that potentially destabilized pentameric interface can cause a release of this intermediate, and that's how we decouple it from catalysis. But we got to make sure that we have robust particles in our reconstruction, and we're following up with other biochemical experiments now to follow up on this and really probe this model more deeply and understand what's going on. And so this leads to now what I'm going to call a working model, because I expect this to go through multiple rounds of editing before we finalize this. But this is that the oligomeric state interfaces can also bias the GS ensemble and modulate KCAT specifically through the E305 loop. So something I didn't want to bore everyone with a ton of different kinetic evidence, but there's a lot more kinetic experiments that went into this. From viscosity studies, we were able to appreciate that product release and loop conformations are both rate limiting for GS, and that this rate limitation is decoupled in our loop variants as well as our pentameric interface variants. And we also have temperature dependent kinetic studies that show that there's actually very little Km impact when we increase temperature, only Kcat, and that this is decoupled in R298A. I am interpreting this as our biasing of Kcat with temperature is that we're directly impacting the loop conformational state, which is partially rate limiting. And so our working model then from putting all of these together would be that the wild type decamer is going to largely follow the same energy diagram that I presented before. H145R decamer is going to be biased away from the active closed state and towards the more open ensemble. We've collected cryo-EM data just in this past December that we just started analyzing on H145R SLAX. We're very excited to hopefully have some structures of that coming out soon. And then for N152G, it seems like even more disrupted energy landscape that might even be having pentamer formations in the mix too, that could be biasing the loop state in addition. Hopefully I'll have more to this story to be able to present at BPS if anyone wants to come chat.
All right,
and
Eric,
then,
I'm going to interrupt you for a second,
Yeah.
just because we are coming up on the hour.
Oh,
So
no.
for those who have to run off, I just wanted to say thank you for joining us. We're going to stick around for another couple of minutes. Eric, I'm not sure how long your last section was and if you wanted to give highlights, or if you just wanted to answer a couple of the questions that have come in. Yeah, we'll stick around another couple minutes and we will post the recording. For those of you who do have to head off, if you submitted questions you're really excited about, come back and find the recording. Otherwise, we'll stick around a couple minutes and hope we see you all again next month. So Eric, did you want to how much is in your last section?
I'd say 5 minutes.
Okay, yeah. Why don't you do that real quick? And then there are a couple of questions, so we'll come to those.
Okay, perfect. Yeah, sorry for going over time here. And so I'm just going to end on a bit of a fun note. So in my new position, able to teach biochem lab classes, and I wanted to develop GS experimentation into that class. So just as a brief primer, so course-based undergraduate research experiences is the acronym CURE. They're increasingly popular and they have very beneficial student outcomes, so increased motivation, increased understanding. Many studies back this up, and there's a huge number of CURE studies that are coming out in the literature now. And the one caveat is that it does require some times and some funding to be able to implement these into the classroom. So my CURE design was relatively simple. How do disease variants impact structure and function of GS? Adapted the course with new protocols, assignments, gave students literature sources, ordered the reagents and prep materials, wrote a small section of a lab manual, and lectured on this project overall to really implement this last semester. This is just an exquisite gel, I think, from the students in the biochem class looking at the R341C variant. This was prepped to near high purity in the class. I could follow this up with size exclusion. And we just really had a lot of fun, like doing some original research in the biochem lab. This is some of the students. They posted this to their B-reel. I learned what that is. And we also, the same non-specific ATP hydrolysis that I mentioned for N152G is also apparent for one of these disease mutants too. So we saw no increase in activity as a function of ammonia, whereas we could get strong Michaelis Menten kinetics for glutamate. And we also have some evidence from mass spectrometry that an R341C has a biased oligomeric state towards pentamer over decamer. And so we're looking to follow up with this. I'm very excited that just earlier this month, I collected data sets for 2 of the disease variants at SLAC. And I have now a master's student who will lead this project and hopefully bring this home. And I just want to conclude by saying thank you for all your patience sticking with me this whole time. I hope I can convince you of a fun story about GS filamentation, the power of ensemble analysis for deciphering enzyme structure, and then hopefully a little bit of an inspiration for any value educators out there for implementing cures in your teaching. I have a huge number of people to thank, and I thanked a lot on the way. Hiroki's been just tremendous in helping drive forward the Ensemble project. My collaborators, Justin and Richard at UW, Max and Sam at Institute Pasteur have been just absolutely incredible, as well as my postdoc mentor, Jamie Frazier, and Angel Arata, who was a technician in the lab at the time, who also helped with the disease project. And all of this is really facilitated by having microscope access at SLAC, and being so spatially or geographically located is very, very great. So I'm happy to take questions now.
That was absolutely great, Eric. Thanks. It's great to see undergraduates in the pipeline for training and for looking at cryo-EM as one of the things they can do. Okay, we have three questions so far from the Q&A. The first is, can you please comment on the role of divalent cations, such as nickel +2 ions in GS filament formation? Is this an alternative mechanism to the one you described?
Yeah, so I think that question might be getting towards, there's also been GS filament formation reported for E. coli GS that I think is facilitated by nickel 2. I haven't studied nickel 2 specifically for human GS. And so I can't comment on that as being a role. I can say that the divalent cations are quite important specifically for the active site. One thing that was a little different revisiting the system compared to some of the really old literature is that like the crystal structure has 3 manganese ions in the active site. We find that manganese actually deeply inhibits the enzyme, and that magnesium is the preferred divalent there. And additionally, that we require 2 magnesium per 1 ATP. So finding exactly what the stoichiometry is part of our other question.
Okay, another question, very nice talk. My question, did you try activity or structural studies with GTP instead of ATP? Is there any specific motif that determines ATP specificity?
Wow, that's a great question. That's something that hadn't occurred to me. I think I was mostly focused on the known forward reaction with ATP. I have not tried anything with GTP. That would be an interesting thing to try.
Okay, another question. Matt McLeod has a similar case where an interface is missing density for a side chain, even though the main chain is well fit. Thoughts on this? Is this highlighting something dynamic? Radiation damage, again, that's something that people have argued about. And how do you go about modeling these regions? So you have an interface missing a density for a side chain where the main chain is pretty visible.
Yeah, so I think that was something that was very intriguing from our reconstruction. So we, based on having immediate secondary structure that was readily decipherable on either side of our loop, made us focus in that it was likely going to be the conformational state of the loop itself. And you can think about it from a statistical standpoint. If you have, say, 50,000 particles in your reconstruction, if 49,000 all have the loop in one conformation, then you're going to really see it. But if only 5,000 are in that conformation, it's only going to be sometimes there, and you can average things in when there's many particles in the conformation, or average things out if there's not many particles in that conformation. So that's where we landed on that. In addition, we were also able to verify this with the time resolved data set that I mentioned too. We also got a full filament reconstruction from that under turnover conditions, which also showed that the loop wasn't there. So having like the secondary line of evidence, I don't think is always necessary, but it was really helpful in just feeling grounded in our results for this story. For the radiation damage, I'll say that, of course, that's like certainly an impact for more of the negatively charged amino acids. And for the ensemble analysis, I think we actually do a little tweak to those to make sure that those aren't being too downweighted for that. But I'd highly recommend reading more about MEVOX and ensemble refinement from Bonomi's group. It's out on bioRxiv now, and the software is open source. So, and they're very, very nice and helpful people if you want to adopt it. I also know Sam is going to be at Biophysical Society this year too, so please seek him out. He'll also be featuring the story there. And so for modeling these, we started with modeling. I like to start with a base model, fix some things with Isolda, go back to Phoenix to refine, go back to Isolda to polish, and then feed what is already a really good model into MEVOX for the simulation-based refinement, which just seems to boost all of the metrics to a level higher.
I have one quick question that is because we are also a cryo-ET center. Is there a possibility of looking for filaments inside cells? That is, could you make a conditions under which you expect to see these filamentous structures, and where in the cell they might be, and how do they find each other in the cell?
Yeah, this is a great question. I think this would be a really excellent extension of this work would be to start doing ET on this. GS is a cytosolic enzyme, so it's going to be just diffuse everywhere. I think some of our, yeah, I should say that there's actually a lot of cell biology too that I haven't presented here, trying to look at growth phenotypes of these different variants, as well as we tried to do an IF experiment to look specifically for puncta. I think it's just a little hard because what we're seeing is filaments of relatively small size. So I think it's I think it'll be hard to identify that. But I actually I think that that would be a very exciting thing to at least put some effort towards.
Thanks. That's all the open questions. Christina?
Yeah. We can go ahead and wrap up then. That was a beautiful story, Eric. These are very hard questions to probe, and so that these are approaches I was not super familiar with, so it's really exciting. And I especially appreciate the way you're using it to drive undergraduate research experiences as well. That's super awesome. I'm glad you shared that.
Yeah, and I can say too, definitely open to any collaborations. If there are folks out there that want to share any data for analysis, I'm hoping to put together a cryo-EM data analysis course. In our new building that we're waiting for, we have a 28 workstation computer lab spec'd out and ready for putting CryoSpark on all of them.
Awesome. That is good to know. I know other ears who may be interested, so I'll pass that on.
Cool.
And I will not be at biophysics, but now I am very jealous. It sounds like it will be a fantastic time.